Hello, I'm David Mead, the CEO and co-founder of Varigen Biosciences. We're an early-stage drug discovery company, and I'm here today with my colleague, David Pompliano, to introduce you to Varigen. The pharmaceutical industry was founded on medicines from natural sources, and over 60% of marketed drugs are natural products or their derivatives. Although natural products discovery has been out of favor recently, the realization that we've looked at less than 1/10 of 1% of the metabolic output of microbes has reignited interest in seeing what other therapeutic opportunities nature might provide. Varigen is leveraging the information gained from microbial genomics to discover new drugs. Our proprietary synthetic biology tools are 10 times faster than existing capabilities, and we identify new natural product compounds as lead molecules for disease targets of high interest to pharma. Our aim is to work hand in glove with therapeutic area experts in pharma in partnerships where we do the compound discovery, they do the pharmacological evaluation of the compounds, and we both contribute to the lead optimization effort. It is time to apply modern synthetic biology methods to natural products drug discovery. Now I'd like to turn it over to David Pompliano.
Thanks, David. Drug discovery is a complicated process, but it all must start with a lead compound, some molecule that shows specific disease-modifying activity in a relevant screening assay. For many programs, the chemical diversity found in synthetic chemical screening libraries is simply not adequate for finding this. As you can see in the figure in the lower left, the synthetic compounds in orange occupy a much smaller portion of chemical space than do natural product compounds in green. Simply on the basis of probability, the greater variation in chemical structure of natural products should provide more opportunities to find hit compounds. The other major problem in early stage discovery is the multifactorial nature of lead optimization. Changes that improve one feature, like say specificity, often diminishes another property, such as say protein binding, not unlike the problem in the whack-a-mole game. So we would like to start with a compound that has as many desirable properties as possible, not just potency in the primary assay. Why screen natural products? Natural products have essentially a billion-year head start in lead identification. Small molecule metabolites have co-evolved to bind and modulate the enzymes and receptors that they interact with. So there's already an inherent specificity for macromolecules that a random collection of synthetic molecules simply don't have. This is shown in the cartoon at the right, this complementarity. Natural products have remarkable structural diversity, multiple stereocenters, various heteroatoms, complex ring structures, and they can interact with a wide range of biological targets. The various properties of natural products are selected in the context of their environment. Evolution provides simultaneous optimization of several traits important for lead compounds, such as selectivity, potency, and localization. With some forethought, we can predict which ecological niche might produce compounds that have properties optimized for human administration. And remember that there are some natural products that are drugs right out of the box. Many antibiotics like amphotericin and erythromycin, as well as FK506, rapamycin, and others. Historically, natural product discovery relied on culturing microbes and testing the growth media for biological activity. If the bug couldn't be grown, its compound output couldn't be tested. However, genome sequencing of the DNA from environmental samples, i.e. dirt, showed that only a tiny fraction of the species that were present in a sample could be cultured. There are literally billions of microbial strains yet to be discovered with the potential to produce all sorts of new metabolites. But now we don't need to be able to culture a microbe to produce the compounds it can make. We can simply isolate all the DNA from an environmental sample, that is, from the metagenome of the sample, identify gene clusters encoding secondary metabolite pathways by their DNA sequence, and express the metabolite heterologously. We can access the biosynthetic potential of virtually all members of the microbial community. So, our vision is to realize the therapeutic potential of all microbial natural products, whether they come from dirt, aquatic environments, or the human microbiome. We aim to go right to the source code in the DNA of microbes for the blueprints to new medicines. Our mission is to be able to make any molecule that nature can make, and then to be able to modify it in ways that nature has not or cannot. Our immediate goal is to produce novel preclinical drug candidates for important disease-implicated biological targets. This means generating a large number of natural products that can be screened in functional assays. Our platform includes multiple expression strains, each of which has a different metabolic background, so that a given clone expressing a biosynthetic pathway might yield several different products, depending upon which native enzymes or alternative substrates are present. This method of augmenting the chemical diversity of natural products is illustrated in the figure below. Once an interesting hit compound is identified, we can optimize it in several ways: by synthetic biological manipulation, by in vitro enzymatic transformation, or by chemical synthesis, in order to produce viable preclinical drug candidates. And now I will turn it back over to David Mead, who will explain the details of our approach.
We are not the only ones using genome information to generate new drugs or agrochemicals. The process has been worked out over the past decade or so, and it is outlined here. As you can see, it can take 12 to 24 months or longer. Starting with the genome sequence, bioinformatic analysis mines specific DNA sequences encoding presumed secondary metabolites. The process of activating the selected biosynthetic gene cluster to produce a metabolite is labor and time intensive, since the gene clusters can be very large and require specialized techniques to manipulate. The biosynthetic gene cluster may require refactoring and many trial and error with different expression conditions to actually make the metabolite. Varigen has developed multiple tools that can dramatically accelerate this process up to tenfold. Varigen excels at this particular task and is a major differentiating factor of our company. For example, using DNA trap to capture a pathway into a novel dual inducible promoter vector technology called BGC Express, we can shorten this process to 2 to 3 months. There are two approaches to identifying new drug leads: a sequence-based and a function-based approach. In the sequence-based approach, the natural product scaffold and the biosynthetic gene cluster encoding it have already been identified, and new congeners of that core scaffold are identified by sequence analysis. Our DNA trap technology is used to clone the BGC from the host organism or from a library of metagenomic DNA and then expressed using the platform as shown in the previous slides. In the function-based approach, it is purely a diagnostic screen of whatever molecules are produced from a metagenomic library. The BGCs from the active wells are then targeted for expression. Varigen has proprietary technology for both approaches. How are we different? Our metagenomic DNA libraries are housed in bacterial artificial chromosomes, or BACs, and are significantly different from those of other companies and academic groups. Importantly, they contain large DNA inserts, with the average insert being over 100 kb. Our big DNA metagenomic libraries contain complete biosynthetic pathways. We know this because all of the inserts have been completely sequenced and subjected to bioinformatic analysis. By this evaluation, there are 449 novel BGCs containing PKS and/or NERPS motifs. We have a second library that is 5 times as large, currently undergoing sequencing of the inserts. Our libraries are different because our vector can capture entire biosynthetic pathways of 100 kb or more. Other groups use phosphid vectors to construct their libraries. These vectors can only contain inserts up to 40 kb, as shown by the shaded green area on the slide. Since they cannot hold full-length pathways, they cannot express the fully elaborated metabolite, so screening for function agnostically with extracts of the phosphid library will not be very fruitful. By contrast, our full-length BGC inserts can and do express novel metabolites that can be screened for drug-like activity. BGC-containing clones were expressed in Streptomyces and then screened for antibacterial activity against 3 pathogens. We found many hits, some narrow spectrum and some broader spectrum, as shown in the Venn diagram on the bottom right of the slide. We are now determining the structures of the active compounds. This is truly natural products discovery 2.0. Unbiased screening of novel compounds produced from the DNA of unculturable microbes. How else are we different? We have multiple proprietary tools shown on the slide, all of which facilitate the production of end-product metabolite from a specific bacterial gene cluster, whether it came from microbial genome sequencing campaign, a bioinformatics analysis, or from a hit and a functional screen of a metagenomic library, like we saw previously. More importantly, we have the in-house expertise and experience that reliably generates final product. For efficient genome mining, Varigen scientists have developed a Cas9 cleavage-based method called DNA trap to specifically cut the DNA and clone the fragment into our expression vectors. The process is shown on the left side. We've had great success in cloning over 100 different BGCs from both bacteria and from fungi, as you can see on the right side. We routinely clone polyketide syntases and non-ribosomal protein synthase gene clusters of greater than 100 kb, and Varigen is only one of 2 groups worldwide that can do this. And we can produce the end product metabolite from the cloned BGCs in our matched vector strain set of expression systems. As you can see from this slide, we can express clones of Bacillus, Streptomyces, Aspergillus, and in various gram-negative strains. This gives us the best chance of making the end product metabolite of a cryptic BGC, especially if it was cloned from a metagenomic library where the original host species is unknown. Producing enough end product metabolite for subsequent structure determination and biological evaluation studies is an ever-present problem in heterologous expression systems. Varigen's proprietary patent-pending dual promoter vector called BGC Express, shown on the right, increases the yields of end product metabolite by 2 to 3-fold or more in some cases. For metabolites like actinirodin and undecylprodigicin with chromophores in the visible spectrum, the advantage of the dual promoter configuration for enhanced expression is self-evident. The BGC Express system was used by Neil Kelleher's group to sort out the biosynthetic pathway of the stravitins. The paper describing the work was just published in ACS Chemical Biology. Here's a comparison of our technology with some of our competitors. Without going into too much detail, our major differentiators are our ability to clone very large pieces of DNA, ones which contain complete biosynthetic gene clusters encoding complex secondary metabolites, our dual inducible promoter technology for gene expression, and our set of matched vector chassis systems from multiple different microbial species. Much of our success in production of end product metabolite
is impressive expertise and experience of our scientific staff. We are often asked to clone or express BGCs when others' efforts to do so have not worked. We succeed where others don't in this area. And now back to David Pompliano for the last few slides.
Thanks, David Mead. I'd like to quickly summarize our presentation today. Varigen is reinventing natural product discovery with our differentiated synthetic biology tools. Our main advantage is our expertise in cloning and expressing the large DNAs that contain complete biosynthetic gene clusters. And our aim is to accelerate the production of cryptic compounds encoded in the genomes of microbes so that they may be screened for pharmacological activity. This unprecedented access to biologically relevant chemical diversity will aid in finding new starting points for previously intractable or undruggable targets. HIT compounds can be modified using various in vitro and in celluloid techniques to improve their developability properties. As the picture at the bottom right shows, Varigen technology is a portal into new chemical space and new opportunities for drug discovery. So, ways of working together. We would like to work in partnership with therapeutic area experts in pharma biotech. As we've shown during this presentation, Varigen can generate and modify natural products from environmental DNA. We can serve as the very front end of a new discovery effort, or we might revive an existing target project where previous screening efforts have failed. If you already have a natural product that you're interested in, we can clone the biosynthetic gene cluster that encodes it, find congeners from the sequence information, and biologically or biochemically modify its structure. In the end, our aim is to collaborate to find and develop new medicines. Our leadership and board have deep scientific, drug discovery, and business expertise. All of the leadership are entrepreneurs, most of whom have started and run, or at least invested in biotech companies. You can see the broad range of the corporate and academic experiences that we have on this slide. Finally, thank you for taking the time to hear the Varigen story. Please contact us if you have any questions about our presentation. We do hope to hear from you. Thanks again.